Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Forget frequently asked questions,
common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Dick Vethaak. He's a researcher, water quality, and ecotoxicology. We're going to talk about the presence of microplastics in human blood and water quality issues that he works on as well. For the rest of his bio, I'll let Dick describe it because there's various Dutch words that I'm probably going to mispronounce. But Dick, thank you for coming.
Yeah, very welcome. And thank you very much for having me. Well, let's say I'm not a medical scientist. In fact, my training is biology and toxicology. I'm not working anymore, an emeritus professor, as we call it. And I'm partly retired for several years now, but I'm still quite active in the field of microplastic research. So,
Why are you active in it? What do you like about it? What fascinates you?
Well, I started a couple of things, before my retirement, and I want just to finish them in a proper way. And I can see that I can make a sort of difference. I have a good network, and it's about your legacy, what you, and I'm not ready yet to stop. And that's one thing. The other thing is, of course, I have a little kid here as well. So if I look in the future of this planet, I think, there's a lot of work still to be done.
Yeah. So what aspects of microplastics are you still looking into? What kind of questions are you trying to answer?
Well, the thing is, I started to look at microplastics, let's say 15 years ago for the first time in the ocean. But then I moved to the fresh waters. There were sewer treatment plants, as potential sources. And then from there, we discovered that these microplastics were about everywhere, also in the air. So in fact, at one point in time realized, that we are breathing in, we eating and drinking microplastics all of the time. So then I moved to the human health side of this issue. We published in 2016 a little paper in Environmental Science Technology stating microplastics debris is a human health issue. So there my journey, my roadmap started, into this adventure. And so we started to look for money, for funding to start researching to this issue, this human health issue. And from there in 2018, this is ZonMW, which is the organization for health research in the Netherlands. They funded 15 breakthrough projects on this issue. 1 year project, but very, very challenging, but very spectacular. And the thing is, the microplastics in blood study you refer to is one of these breakthrough projects. So there are several other projects as well that gave quite some interesting results. Now, these results were already available for a while because in 2020, I think we started to set up this consortium, this Momentum Consortium with 27 partners, including the public and the private sector, but also I think about 10 different universities and other research organizations in the Netherlands. So with about 5.5 million euros, we started this 3-year project I think we were a front runner in this respect in the Netherlands, looking at this potential health issue. And so that was my adventure. I became co-coordinator of this momentum, and it's still running. And at the moment, I'm even now looking for money for follow-up, because I can see, this is not a question of 3 years research. I think we probably need 10, 15 years of research to tackle some of these problems.
Well, it's a new field, and a lot of people have questions. Tell me about the microplastics in blood research. I don't know how closely you're involved, but what kind of things did you study, and what did you find out?
Well, that was quite something. We started to look at that as a team at the Vrije Universiteit Amsterdam. I'm just one of the co-authors of this paper, so I'm not a chemist. But the work was published in Environmental International this year, and the social media they went crazy over it. The article has already been cited more than 300 times in half a year time. It's amazing. So
it's kind of crazy to have microplastics in your blood.
It is.
The first thing it made me think of, thing it made me think was that people with leaky gut syndrome probably are a lot more prevalent to it. But that was just my speculation.
Yeah, this is speculation, but I think we expected something like that. We knew it already for many other animals, like aquatic or animals, but also rodents. That if you expose them to micro-nanoplastics, these particles are rapidly taken up in the circulation, and so we could expect something like that. Particles can enter the blood system, the circulation in humans, as but what's a bit surprising, the size of the particles, the upper size limit. So
what were some of the sizes, and what was unexpected? They were much bigger than you'd expect, or what?
Yeah, we still don't know exactly what the upper size limit is, but usually we know about nano-sized plastic. They can take these barriers, but we find particles as large as 20 micrometer that can cross these epithelial membranes, for example, and so that can enter the blood system. So it's quite a new field. What we have done here, we developed a robust and a sensitive method for polymer mass concentrations in blood. So we used what we call a double shot gas chromatography mass spectrometry method. And that means this is a destructive method. And maybe you've heard of other studies, that reported on microplastics in lung tissue or in placentas. They used a different method. They used spectroscopic method. So they looked at the particles themselves and the particle sizes. We used the destructive method. So we destructed using pyrolysis. We destructed in several steps the plastics. Then we ended up, quantifying the thermal products of the plastic particles in the samples, and then we come up mass concentration rather than a particle number. So these are different methods, and it's
you destructed stuff by pyrolysis. Were the original polymers preserved, or what broke down, or what stayed?
Well, yeah. Well, initially, the volatile compounds like the additives, etc., they are thermally extracted from the sample at relatively low temperature, I think 200 degrees. And then the next step is the same sample paralyzed at a higher temperature. And then you destroy the polymers themselves. It's a semi-quantitative technique that quantifies the thermal degradation products of the plastic particles present in the sample. And so,
what are you able to quantify if do you do like low temperature pyrolysis, so the molecules are still preserved, or at what temperature do they all just essentially combust or break down?
I think it was 400 degrees.
Are you doing it in the absence of air? So again,
in absence of air, yes, of oxygen, okay. Of oxygen.
Oxygen.
Yeah.
Okay. So first of all, some of the surprises were that you had pretty large microplastics, like you said, up to 20 microns.
No, yeah. Well, I said that, that is just coming from other studies where we used in vitro experiments, with animal and human cell lines. And we find a rather high, but I said, large particle sizes that are capable to cross these barriers. But that is a different story. But it has something to do with this as well, because, if you only have a mass, and we determined for the first time mass of plastic particles in blood, then that could be attributed to maybe a couple of larger particles, or maybe many, many small particles. We don't know. The other side of the story, we need other complementary methods to say something about the particle distributions and particle size distribution, etc., etc. Probably also the shapes. And this is something that is still in the pipeline.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now, back to the show.
Why don't they take the blood and maybe centrifuge it down and, then run the remaining plastics through, a gas chromatograph if they can? Like, what would they need to do? I mean, well, again, if you spun down the blood, if you centrifuged it, I would guess you could probably separate the plastics out of it, but then how would you analyze them, while preserving them?
Yeah, well, it's not so easy. There is another key issue in this type of analysis, and that is the quality control of the background plastics that, throughout the sampling and analysis is very key. I mean, we know that everything, micro nanoplastics are about everywhere. So if you take your sample, extract blood, it can be very easily contaminated through the air, from the air, or when you do the analysis in a certain room and you wear clothes, that can release fibers, etc., that can contaminate your sample as well during the analysis even. So you have to be very careful with that. That's why we use blanks, in all these steps. And that is something that is commonly used, in this sort of analysis, chemical analysis. You use blanks and you extract the amounts of micro nanoplastic you find in your blanks. You extract that from your findings in your blood sample.
Because microplastics are everywhere, a lot can fall into the sample, I guess, or abrade off of the lab equipment.
Yeah, same thing, So the thing is, you have to realize, if you take blood, you should use no plastics. You should
or people's clothing, or I guess there's a lot of sources of it. Yeah.
That's why you use these blanks, So blanks means, you use a meter disc there, and see how much is depositing coming from the air, how much is the fallout on this petri disc. And so you can have an idea about the potential contamination, cross-contamination. But having said that, there's still a lot of work to be done, because this is a pioneering study. We took samples from 22 healthy blood donors from the general population. And again, also the size range captured is determined, the diameter of the sterile stainless needle, which was 0.5 millimeters. And the lower limit was determined by the glass filter mesh size of 700 nanometers. So we only looked at a particular size range within these micro nanoplastics class. But also, we looked only, let's say, at only 5 or 6 target polymer types. So we looked at a couple of commodity plastics. So again, It is just the first step, but there's much more work to be done because we are exposed to many more polymer types than just the 5 target types.
What are some last conclusions that came from those experiments that you read? Did they appear to prove that there was a substantial amount of microplastics? Did they, were any of the experimenters able to clarify what kind of microplastics and how many?
Yeah, so what we found is we targeted these 5 high production volume polymers that we use in daily life, like PET, PE, polyethylene, polymers of styrene, etc. I think these were the most widely encountered. And then we also found, I think in some of these donors, we found polymethyl acrylate. And in none of these samples, we couldn't find any PP, polypropylene particles. So if you look at the mean sum concentrations of these plastic particles in blood, which was 1.6 microgram per microliter, that is quite high, if you compare that to river water or whatever. So we don't, at that point, we say, we proved that there is plastic particles present in the blood, but we have to confirm these these concentrations, it's just the very first study, very small numbers of donors we looked at. And we need to extend the numbers of donors and maybe also the number of polymer types and also further probably, to mature the method. And that's work is now already underway, in this Momentum consortium.
All right, but okay. So, what? Any other experimentation that you read about or you were involved in that just amazed you or blew you away?
That blew me away. Well,
surprised
for me, I mean, I'm pretty into this field, but I was really surprised. For me, this type of research is basically putting different disciplines together. I'm an environmental toxicologist. And now I work with medical specialists, medical experts, scientists. They have their own methods. They have their own ideas. And if you put them together with our ways and our methods, you see you have a win-win situation because there's so much you win from working together. And that's exactly what opened my eyes. I think 2 years ago, I was asked by a group in Austria, and this is a medical hospital, veterinarian and medical hospital in Vienna. I was asked to advise them on a microplastics research proposal. They were one of the first there as well. And the thing is, they were focusing completely only on the carcinogenic effects of particles, microplastic particles and nanoplastic particles, carcinogenic effects, which we never realized, that could be an issue as well. But now we have a team there of oncologists that are focusing particularly on, this is for me a very exciting thing, That is one thing. And another example is, which I find very intriguing, is these particles. As you know, we're not just exposed to these particles, we're exposed to both particles and these additives that can leach out from these particles. But there is also this issue of the corona formation. Once in the environment, the surface, it will be covered with what we call an ecocorona that contains natural matter, but also biomolecules like lipids, for example, and chemical and microbiological contaminants, etc., etc. And this corona on the surface of this particle is depending on the surrounding conditions, of course, in the environment. And that adds significantly to the complexity of the micro nanoplastics, because they play quite an important role, I think, in the fate and toxicity of these particles. And you see a lot of experiments that have been done now with micro nanoplastics are done with virgin, primarily the polystyrene particles. And now we see more and more results with wetted microplastics that contain this corona, where we find more positive results. So positive results in terms of potential health effects. So that is quite an intriguing thing because this corona is changing, the composition is changing if you go from the outside to the inside across organs. And it's very complex, but it's for me, very intriguing, exciting.
Well, okay. So they've identified that like coronavirus will take harbor on microplastics? Is that what you're saying?
For example, there are some studies that suggest that the COVID virus could be transmitted through plastic aerosols, for example.
Well, in general, I mean, do these microplastics have their own unique microbiomes? With various bacteria?
They have distinct microbiomes. Yes, they have distinct. That could also include pathogens, human pathogens.
Oh, pathogens. Okay, I'm sorry, I couldn't understand. What have you seen from looking at the microbiome associated with microplastics? Is it has anyone done metagenomics to see the function of the bacteria that are on the microplastics? So they're using it as a substrate and eating it, or what seems to be the function of the microbiome associated with it?
Well, so far as we understand it, these microplastics upon ingestion, they could influence the gut microbiome. There are some studies that suggest that. And of course, you have to realize that these microplastics that contain these biofilms, they have to cross also the harsh conditions of the stomach.
Other research projects have you again looked at that I don't know, you feel like really informed you a lot about the future consequence of microplastics?
Other projects where most of these projects I'm partly involved in are in vitro work, where we use human cell lines to understand, this corona formation and let's say the uptake and translocation of these particles through internal and of course external membranes. And that is one thing, but it's quite exciting work going on. The thing is, you need to understand the internal doses. First of all, the internal doses to say something about, to determine the risk. I mean,
what would constitute I guess we don't even know how much, again, the microplastics load that people get in different situations.
No, we don't know yet.
For certain ones, it's incredibly high, some low.
No, we don't know yet. The thing is, we need to look at we need biomonitoring studies where we look at the general population. And that means that if I drink from a paper cup now, that is filled with, hot coffee, these paper cups, they have an inside on a lining of polyethylene. I'm exposed to, when I drink it, I'm exposed to vast numbers of micro nano PE particles. So these very high acute exposures, they happen all the time. Also, when you cook, in using plastic, like these pans that have these plastic layers, the Teflon layers, they release, if you scratch them or damage them, they release a huge amount of plastic particles that you inhale, etc., etc. So there are acute exposures of very high levels, but in general, we are exposed all of the time to these particles, lifetime. And the thing is, if we accumulate these particles, and we're not sure yet if they accumulate in certain organs, if they accumulate in our body, then obviously I have much more of these plastic particles in my body. I'm almost 70 years old now than you. You see what I mean?
Well, has anyone
around there for decades already? These particles.
What about an experiment where people would, just drink and eat and hang out and then urinate, and they would look at the urine, like the exact example you gave.
Yeah, that could be one.
See what their baseline is. And then they drink, let's say, a hot coffee in a cup, and then does it go up suddenly? Like, how fast are microplastics cleared from the body? They looked at, feces and urine.
That is exactly, you need to know about the kinetics, how much is retained in the body, how much is excreted through urine or the bile, etc. And these studies are underway. I've not seen any study yet looking at micro nanoplastic in urine or looking at these kinetics in such a way, that we can understand how much goes into the body and how much goes out and how much is retained in certain organs. Because
even if you didn't know what the load was, but you saw that someone had a certain amount, let's say they kept 24-hour period and, and then you put them in a situation. Yeah.
The thing is, what we understand now is that most of these plastics you pull out again, and that most of the plastic you inhale, you cough out, and you swallow them, and you put them out as well. So the thing is, we understand so far that it's only a very small fraction of particles that are actually capable of crossing these membranes, the external membranes in the gut or the lung, and so potentially reaching the circulation. Maybe it's less than 1% or something like that. We're not sure yet how much that is. Depends on the polymer types. I think maybe also the corona formations, etc., etc. So the thing is, we probably the very small, the nano-sized plastics, they are more easily taken up than the larger micro-sized plastic or the smaller micro-sized plastics. But there is still a lot we don't know about this. The only thing what we understand now is that these micro nanoplastics they exhibit similar health effects as we know already from well-studied other insoluble particles, like black carbon, for example, or soot particles. And so that is the only thing. And then the question, of course, is do they, as plastic particles, do they have a similar toxicological profile, or maybe are they more toxic than these other ambient particles? Because we live in a multi-particle world. We're exposed to all these particles. particles, the different particles. And now suddenly we have a new particle type here, plastic particles. And so, in that sense, it's also so that you cannot say, microplastics are causing a disease. Now, if they do, they contribute maybe to certain diseases because they're multifactorial. There are other particle types as well that do the same thing. And it is something what I already said at the start, that will take maybe 5 to 10, 15 years research to understand what's going on here. And that's why, all the time I try to plead for this precautionary approach, what are we doing here?
I know a lot of additives are added to, plastic bottles, plasticizers, flame retardants, colors, things like that, to a lot of plastic items. So when plastic breaks down, it becomes, let's say, microplastics. Is it mostly backbone of the plastics that are left, or a lot of these additives still left, interwoven between the molecules of backbone of plastic?
Well, that's a good question. I think that most of the additives probably leach out already in the first weeks, or months when they are in the environment or in the water, for example. But we know that some of these additives are compounded in the polymer matrix. So they're really also in the center. So they can be released, during this process of degradation, where they fall apart in smaller and smaller particles. And that is the point that worries me the most, that if you look into the future, then we are dealing here with a very, black scenario, First of all, that the number of particles will rise in time. We know that. And because there's so much plastic around already in the environment and everywhere in our households and offices, that will degrade. That will break down in smaller and smaller particles over time, constantly rising these micro-nanoplastic levels, that we have to deal with that in the first place. But then there is still a lot of plastic debris and plastics to come in the coming years. You can see we need legislation here to stop the flow of plastics. Otherwise, we are dealing with increasing levels, exposure levels. And in my case, I think, I personally think, with a health emergency in the future of this type of plastics. And so that's why that keeps me also going, I can see, there are some things you can do about it on a personal level. But if you look in the world now at the moment, all these crises, biodiversity, air pollution, climate change, I think plastic is only, has quite some relationship with all of these other crises as well. I think we have to combine forces here and to see, what's happening. We need legislation. There's some work done already now in the European Union. There is this plastic treaty in Uruguay, I think. It was, but it's very slow progress. So slow progress, That worries me.
So, okay. What do you think is going to be the, I don't know, I guess the future of microplastics over the next 5 or 10 years? It seems like, the progress is like a snail's pace. I don't know how society will work without the omnipresent plastics that are just everywhere. But what do you think is going to happen over the next few years?
Well, what I hope is that, that we will phase out most of these, let's say the bad plastics, the most ugly ones. So we have to identify these and then replace them with, more eco-friendly materials or plastics. I think plastic will still be the future somehow. I can see that, but we have to find out, first of all, what are the bad and the ugly guys and what are the good guys. So we can replace some of these, of the most ugly ones. We have to reduce the amount of plastics in the whole system. So, and of course, look for alternative materials at the same time. And it's not easy, because you want, if you look at these companies, we talked about these additives. For many of them are endocrine-disrupting chemicals. There's a similar issue there that is already 20, 30 years going on, and there's still a lot of opposition. There's a lot of lobby by the industry, to keep these endocrine-disrupting chemicals alive, and use them in all sort of products. And this is a similar story. So I'm a bit pessimistic if I think about the future. Legislation. I think I've never seen any environmental problem solved without any legislation. So the industry should maybe pay taxes, et cetera, and all these incentives that may work. But let's see what happens. again, we have to see how much plastics are really inside of our bodies. Do they accumulate? And what are these threshold levels where we can expect diseases or contribution to diseases like, that can also be linked to all sort of, chronic diseases like degenerative chronic diseases.
What's the best way for people to find out more about your work? You said you had a substantial network. So where can people go? Do you have a website that people can look at, or where can they go?
Yeah, there are websites they can look at. What I can say, you Google Dick Vethaak plastics and you find all the items on the internet and the articles. We had an article out, I think, a couple of months ago in Think Global Health. And that article gives you a sort of summary of the state of research and the potential health emergency we're facing. So that could be a good start, to look at that particular article. But of course, we have this Momentum project in the Netherlands, which can be found on the internet. And there are a lot of articles. We had the article in Science. We're not the only one. There are many other scientists that produce excellent articles and even provide information about this plastic nanoplastics health issues.
Okay. Well, very good. Dick, thank you for coming on the podcast. I appreciate your dedication to this field, even though you're formally retired, but you're still working on it. So again, thank you for being here.
You're welcome. Take care there.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.